Okay, so we have a ton of practice with this protein synthesis stuff because I feel like it's a little bit, it's a little hard to grasp. And so we have lots of practice problems to do with it. So let's go ahead and get into it, and I'll go in through the answers now, but you guys can do these on your own first. So first practice problem, what are the 3 types of mRNA we talked about and their functions? So the first one is rRNA, and it forms the ribosomes. mRNA is the messenger RNA, so it's like the code. So it transfers code to ribosomes. And then the last one is tRNA, and it's going to be our transport. It's going to transport amino acids for protein synthesis. Okay, write out the sequence of mRNA that would be synthesized from the following template DNA strand. So if I'm making mRNA, I have to remember RNA has U, not T. So we have our 5' end of our DNA, 3' end of our RNA. I'm sorry, 3' end at the other end. RNA would be 3' down here. Doesn't necessarily have to be mRNA, it just specifically says that in the problem. So I'm gonna have mRNA, I'm gonna zoom in just because these letters are small. The opposite of T is going to be A. Opposite of A would be T if it's DNA, but since it's RNA, we have U, G, C, A, C, A, G, G, A, G, A, G, C, A, G. And then we have our 5' end at the other side. Okay. So that's the answer for that one, how we would get to our mRNA strand. It's the complementary base pairs, making sure we use the correct kind of bases. What amino acids do the following codons code for? What are the anticodons for each of the corresponding tRNAs? So first, let's look at the protein. So I'm going to draw it on the right. I have my chart in front of me so that I don't have to go back and forth to the earlier pages. This first one is AAA, which is based off of my chart. It looks like it's lysine, so it's LYS. CCU is going to be proline. AGG is arginine. And the last one is GUG. GUG is valine. So this is going to be just the inner part of a protein. It'll have more other parts on the other sides. Okay, and then what are the anticodons for each of the corresponding tRNAs? And so the anticodons are going to be the opposites again. And so they're going to be RNA as well, and then the corresponding base pairs. So the tRNA, it would be, instead of AAA, it would be U, U, U. Remember, we're in RNA, so U and not T. Then we would have G, G, A, U, U, C, whoa, C, 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 A, C. Okay, so those are our anticodons. Given the following DNA strand, what would be the mRNA sequence? What would be the tRNA anticodons? What amino acid sequence is coded? So we have DNA here is what we're told. given the following DNA strand, let's start with red. We have D, N, that's an M, N, A strand. What would be the mRNA sequence? Okay, so we'd have 5' to 3'. So let's do our mRNA in orange. Okay, so we'd have our 3'. We'd have a U because we're in RNA now. C, 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 A, U, A. Then we'd have our 5'. Okay. Beautiful. Okay, so looking in order to get to the tRNA anticodons, it's actually a little bit tricky because we do those anticodons 5' to 3'. So I need to swap the order of the sequence. So the same mRNA strand, now I'm going to write it 5' to 3'. So I'm just going to swap the order. So A, U, A, C, 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 U, A. Now we have 3'. So you see, I just took the exact same order, mirror image kind of thing. So in order to get our correct anticodons, we need to start at the 5' end and then make our anticodons. So let's do a different color for tRNA. So then our tRNA anticodons, we have our 3' here. And we'd have U, A, U, G, 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 A, U. So you'll notice, other than it being swapped the other direction, it's pretty much the same as the DNA portion, but now we're having U's instead of T's, and it's flipped a little bit backwards. Okay, so continuing on. Oh, what is the amino acid sequence encoded? So we're going to look at the codons for that. All right, so these here are the codons, and we need to start at the 5' end. So we're going to look at this one here because we have to start protein synthesis is 5' to 3'. Okay, so we're going to look at this sequence and go back to our chart for our codons. And AAU stands for ASN. Then we have AAC, which is the same, ASN again. CCC is pro, PRO. And then our last one, CUA. CUA is leucine, LEU. So that is the amino acid sequence that this codes for. Okay, we've got a lot to do, so let's just keep chugging along. Match the following terms to a definition or a concept. Ribosomes move along mRNA, adding amino acids and growing a peptide chain. So this is adding one by one to get our peptide chain. This is going to be translocation. Actually, let's just put numbers here. Or letters. This is A. Okay, a completed peptide chain is released. That is going to be termination. That's when we finish making our peptide chain. tRNA attaches to its specific amino acid. So this is the attachment before to its amino acid before it can come meet up with the ribosome and the mRNA. There we go. So that would be our initiation step, C. And then a tRNA binds to the AUG I'm sorry, not initiation, it would be activation. I said that in the middle of my sentence. tRNA binds to the AUG codon of the mRNA of the ribosome. So the AUG codon is the start codon, so that is initiation, D. Following DNA template strand is used to build mRNA for a protein. So what is the corresponding mRNA sequence? Okay, so we're going to start with our 5'. So the same thing we've been doing where we just draw it underneath. I'm just going to write it a line later. But we have used the RNA sequence that we're going to flip and do our complementary base pairs. So we have the opposite of G is C, and then U, because remember we have U in RNA, and then G, 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 A, A, A. And then we have our 3' end. What are the anticodons for the tRNAs? So we have it in the correct order this time. We don't have to flip anything around. So what are the anticodons? So they're going to be now the opposite, again, of those mRNA codons. So our 3' end of our codon, then we have G, A, C, 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 and then U, U, U instead of T, T, T because we're in mRNA. Now we're back to the 5'. And then what is the amino acid order for the peptide? So again, remember, we're going to look at our codons, not our anticodons up here. So let's go to our chart. CUU is leucine, LEU. GGG codes for glycine. And our last one is AAA, which codes for LYS, lysine. Okay, so that finishes up that question. We're on our last one. Place the following statements in order of their occurrence in protein synthesis. So what's first, second, third, fourth, fifth? So the first thing that happens is that DNA produces mRNA. So this is number 1 down here on the bottom. Number 2 is that the mRNA is going to travel to the ribosome and attach to the ribosome. So that's the first one here, number 2, or A is number 2. Okay. After I read all of them. Okay, so the next is that the tRNA is an amino acid, or it brings an amino acid to its codon on the mRNA. So that's the third part, is the amino acid through the tRNA gets transferred to where the ribosome is. Ribosome moves along the mRNA to add amino acids to the growing peptide chain. So that peptide that came in the tRNA gets added to that, step number 4. Lastly, a completed polypeptide chain is released. So that's number 5. You release your final protein chain. Okay, so that was a lot of practice. I hope you guys got good practice out of that and understand the material a bit better.